Coming up next, we have Dr. Thomas Guttuso talking about research.
Now it's working. Good. So yes, we don't want to forget Mother's Day, do we? So happy Mother's Day to everybody. Those of you who know me know my favorite pastime activity is kite surfing. And if you remember yesterday, it was kind of windy. And fortunately, I was able to get out on Lake Erie with some friends and do some kite surfing. And one of my buddies, after we were done, said, you know, it's supposed to be windy on Sunday. You coming out? I said, well, you know, Bill, it's Mother's Day, and I need to spend some time with my wife. And he said, well, she's not your mother. I said, now I know why you've been divorced twice. So let's talk about some latest research, some unfortunate news with some trials we've been waiting to hear on, and then some interesting, exciting news. Last year, I talked to you about a nicotine patch trial that really flopped. I'm going to talk a little bit more about smoking at the very end of this presentation. So let me see how I advance here. Green button. So I'm going to be just talking about research focused on ways to slow down Parkinson's progression, what we call disease-modifying therapies. I'm not going to really be talking about symptomatic therapies, ways to treat your motor symptoms or non-motor symptoms. And so there's been a lot of research going on focused on this, alpha-synuclein. You may have heard about this protein. This is what accumulates in the dopamine cells in the brain in Parkinson's, and we believe is one of the main reasons these cells become dysfunctional and start to die and dopamine levels drop, and then people get the motor symptoms of Parkinson's. And so alpha-synuclein was kind of first discovered from families that had a defect in the gene that codes for alpha-synuclein. Their alpha-synuclein levels went way up, and they would develop Parkinson's. These Lewy bodies that are trapped inside these dopamine cells, we've known about since the early 1900s, but over like the past 15, 20 years, there's been more and more evidence that this alpha-synuclein may be one of the most important targets to try and clear out of the brain and maybe even clear out of, we heard Dr. Cloud talk about the gut, the nervous system that innervates the gut. So a lot of companies are focusing on alpha-synuclein, and some of the repurposed drugs that I'm going to be talking about are based on their ability to clear out alpha-synuclein inside brain cells. So this is that Brock hypothesis that Dr. Cloud referred to. And so this is what we believe is what's happening in Parkinson's, is these alpha-synuclein aggregates, these clumps of protein. They start, initially, you can see, I don't know if there's a laser light on here. Is that the red button? No? The green button. Aha. So you can see, initially, these alpha-synuclein deposit down here in this medulla part of the brain stem. And even in this little tiny part peeking out here is the olfactory bulb. That's the part of the brain that senses smell. And we heard that about 90% of patients with Parkinson's have problems with their sense of smell. Actually, just a show of hands, how many Parkinson's patients here feel like their smell is not so good? We got a lot of hands up in the air. And so out of those patients whose sense of smell isn't so good, do you remember how many of you noticed that your sense of smell wasn't right even before you were diagnosed with Parkinson's? Quite a few hands go up again. Probably about 3/4 of you raised your hands that did the initially. And so this is an area of research, because not only does loss of sense of smell predate an actual diagnosis of Parkinson's disease, but like we heard Dr. Cloud talk about, constipation can as well. Mood disorders, the sleep disorder that we heard Dr. Colley talk about, REM sleep behavior disorder. These are ways that we can start to predict that people may go on to develop Parkinson's. And right now we don't have a way to prevent it, but that's where the field is moving. And it's focused on alpha-synuclein. And so as this alpha-synuclein starts to propagate and starts to accumulate, it now works its way into the dopamine part of the brain, which is in the midbrain right here. And this is the symptomatic phase. But according to Brock, that's already at stage 4. So the disease started decades, most likely decades before we actually make a diagnosis of Parkinson's disease. And as the disease progresses, like we just heard Dr. Colley talk about, sometimes these abnormal proteins can move up into the higher part of the brain, the cortex, and then we can develop cognitive problems and sometimes a lot of gait and balance disorders, freezing of gait. So in terms of research, a lot of industry is very interested in immunization therapy. So alpha-synuclein is a protein, and we can design antibodies that glom onto and attach to any particular protein at all. We can design these antibodies to stick to proteins and then hope that our own immune system will clear these proteins, say, out of the brain. And so there's active immunization strategy that it's going on. And so this is just a vaccination. It's a shot, just like you'd go to the doctor to get a vaccination. This is an alpha-synuclein vaccination. The company is called Epheris. They're entering phase 2 clinical trials. Let me just briefly describe the whole drug development process. Most therapies start in animal studies. We show some benefit in animal models of Parkinson's. We show that it doesn't look like it's causing any toxicities in the animals. Then it moves into human studies. Phase 1 is mostly safety and tolerability. Sometimes it's in healthy patients that don't have Parkinson's. Sometimes it might, some patients with Parkinson's might be enrolled in a phase 1 clinical trial. And so that's just to make sure it's safe in humans. And then if it's safe, now it moves into phase 2. Phase 2 is a larger population of patients in the disease category you're interested in, so in Parkinson's. And you're looking now both at safety but also at efficacy. So you'll have a placebo arm in the study. Patients will be randomly assigned to a treatment and to a placebo arm. That's the first time that we start to get a clue, is this therapy effective? Is in phase 2. So this vaccination is now in phase 2 clinical trials. We don't know if it's effective, but from phase 1, it definitely appeared to be safe. And there's a lot of companies working on alpha-synuclein antibody infusions. The two main ones is this Prothena and Roche. This is the name of their drug, prasinezumab. I think I got it right. So it's in phase 2 right now. And so this is an infusion. It's not a vaccination where patients will go into the office once a month or once every 3 weeks to get an infusion of this antibody that's specifically designed to target alpha-synuclein. And another company is Biogen, and this study is also in phase 2 clinical trial right now. BIB54 is the name of theirs. It's a lot easier to say than the other one, which I won't try again. So we don't know if these therapies are effective at this time. Another one that's kind of interesting is an oral medication. The name of the company is called Neuropore Therapies. This is a pill that prevents clumping of the alpha-synuclein. So we know that alpha-synuclein just becomes problematic once it starts getting sticky. And once they start sticking together, it forms these things called oligomers. The oligomers are clumps of alpha-synuclein. Those are the things that are most toxic to nerve cells. And it's a very similar story in Alzheimer's disease. It's just the protein in Alzheimer's is called tau. And as these tau and alpha-synuclein oligomers aggregate, they start to cause a lot of havoc within the central nervous system, the brain causing the symptoms. So Neuropore Therapies is looking at this molecule, NPT 211, that prevents the clumping of alpha-synuclein. And so it was safe in animal models. It's entering phase 1 clinical trials now. So now let's talk about repurposing old drugs for Parkinson's. Last year, hopefully some of you remember a silly little video that I played. It was a clip from one of my favorite movies, Spaceballs. I don't know if you remember. Anybody remember that clip? Some people remember the clip. Where Rick Moranis was Dark Helmet, and he had a head trauma injury, and then when he woke up, he said, "Smoke them if you got them." So that was the introduction to the nicotine patch trial, which was not successful. There's many repurposed drugs that have been funded by the National Institutes of Health as well as Michael J. Fox Foundation. So let's talk about them. I'm going to talk about two that unfortunately did not work out so well. Isradipine is a blood pressure medication. Many of my patients will ask me about these studies, like, these drugs are available right now. Should I ask my doctor to get them? And I tell them, no, let's wait to see what the research shows. Even though some of the early studies look promising, let's see. I don't want to expose you to risk if this is a waste of time. Let's see what the study shows. So isradipine is a blood pressure medication. It targets calcium channels, dopamine cells in Parkinson's that degenerate. They have this pacemaker function, and they're kind of overactive. And if you use these calcium channel blockers in animal models, you can protect dopamine cells. There was also some epidemiologic data that people who were on these medicines were less likely to develop Parkinson's. So that was kind of the background that led to this big study, a phase 3 clinical trial called Steady PD, 336 patients enrolled. And the study, just a few weeks ago, we found out that there was no significant benefit between the placebo group and the isradipine group. We'll get some more data out of this soon to see if maybe there's some subgroups that responded, because Parkinson's is a very heterogeneous disease. And so, maybe thinking about a silver bullet, one medicine that's going to work for everybody is not a good way to think about how to treat patients with Parkinson's. So there'll be more analysis of the isradipine study to see if it's effective in subgroups. Inosine was a very interesting story. So this is a precursor. This is an over-the-counter supplement. You don't even need a prescription for inosine. And the body converts inosine into urate, or uric acid. And if your uric acid levels get up too high, anybody know what you get? Gout. Oh, everybody's very educated here. Gout. Maybe some of you have had gout. Gout is quite painful, especially in the big toe. So that was shown, I'll show you some slides, that there was some evidence that patients with high uric acid levels were less likely to develop Parkinson's. If you had Parkinson's, you had a slower progression. We know uric acid is an antioxidant. And so there was a lot of good evidence that maybe increasing your uric acid levels if you have Parkinson's could slow the progression of the disease. And this worked its way into a phase 3 clinical trial, 270 patients, called SURE-PD3. And this study was stopped early back in December due to futility. There wasn't any chance that there was going to be a difference between the placebo arm and the inosine arm. And so, again, this study, we're going to be looking at some subgroup analysis to see if maybe there were particular patients that responded really well, but the overall group of Parkinson's patients, there wasn't a significant benefit. And so I just want to show you a little bit of the background for the urate trial. I guess it's going to be hard to see this, but this was from a really big Parkinson's study that enrolled 804 patients. That's a really big study. It's called the PRECEPT study. And so when they looked at the patients at baseline, these Parkinson's patients, when they enrolled, the patients who had the highest levels of uric acid at baseline had the slowest progression. They're the green line at the bottom there. While the patients with the lowest amounts of uric acid, they had a faster progression. They're up here. And then the very first safety and tolerability study showed that when you gave patients inosine, let's go back. This is the placebo arm, and this is the rate of progression in their motor symptoms over 24 months, so over 2 years. You can see that the different doses of inosine, it sure looked like at a lower dose of inosine, that looked like it was doing something, and even a higher dose of inosine looked like it was doing something more, less progression in motor symptoms. But again, this study only had 75 patients in it. This was a early stage study. Well, the bigger study that we just reviewed did not show a significant difference. So hopefully, both in the isradipine study and the inosine trial, we'll find subgroups that were responsive to these therapies, because they both had some pretty sound preclinical and early stage clinical evidence of benefit, but the later stage studies did not. So I guess that's some of the bad news. I started with the bad news first. Now let's move to some other repurposed drugs. Nilotinib, I'm sure a lot of you heard about this cancer medication. It's used for leukemia, an FDA-approved medicine for leukemia, and in low doses was showed to be effective in animal models of Parkinson's. This does target alpha-synuclein, and it helps clear alpha-synuclein from the brain. Promising phase 1 clinical data. Now, a very small study, only 12 patients, but did appear to be safe. And this then stimulated 2 phase 2 clinical trials that are going on right now. Nilo-PD, enrolling 135 patients, and PD-nilotinib, enrolling 75 patients. So these are ongoing studies. These are the first studies to test if nilotinib has benefit in Parkinson's over placebo. Finally, exenatide. This is a diabetes medication. It's an injectable medicine. It's not insulin. It has a different mechanism of action. And exenatide, again, has shown benefit in animal models of Parkinson's. It has been shown to clear alpha-synuclein. And in a phase 2 clinical trial, only 62 patients, this was just published a few months ago, was positive. Yay! So, 3 exclamation points and a question mark. So why the question mark? So this is exciting. This is like our first repurposed drug for Parkinson's that we got positive results. Why isn't there much more excitement that you guys have all heard about on, all the web pages and newsletters that you read, that you get? This is why. So if we look at the data, if you just look at the second figure, let me go over here to this figure over here on the right, you can see the placebo arm is the blue, and the exenatide arm is the blue, and the placebo arm is the red. And this is over 48 weeks. Patients randomly assigned to exenatide had this big improvement in their motor symptoms, and then they kind of stabilized. They didn't really show much more improvement after 12 weeks. And at 48 weeks, everybody stopped taking their therapy. Whether you want a placebo injection or you want exenatide injection, everybody came off. And then the final endpoint was at 60 weeks, 12 weeks after you stopped the therapy. And the theory was, if exenatide is actually slowing down the disease and protecting brain cells, you should see a significant difference at the end. This exenatide group shouldn't just catch right back up to the placebo group. And as you can see, they didn't. It was a positive study. This was a significant difference at 12 weeks after stopping therapy. And then when they were on therapy, is even a greater difference between the 2 arms. So what's the holdup? Why doesn't this prove that exenatide is a disease-modifying agent, that it's protecting brain cells, and that we should be actively getting this into bigger trials and getting this into the hands of Parkinson's patients? So the bottom line is, it might, but it might not. This is a problem with how this trial was designed. This trial did not have biomarkers in the study. I'm going to talk about that with a second, but this pattern of benefit is very similar to what we'd see if we did a trial with, say, levodopa or any of our dopamine medications that are already FDA approved and that we know do not slow down the progression of Parkinson's. You get a big symptomatic improvement early on, the improvement plateaus, and then if you stop the medicine, patients will get worse. So it was kind of surprising that exenatide gave us data that looks very much like it just has symptomatic benefit. Nobody ever expected that. We expected that if it was going to work, it would be a slow improvement, just like a steady line going down. Just like the placebo group is kind of very steadily going up, getting worse, that the exenatide group would be very steadily going down the other direction. And then when you stopped it, you might get a little bit of worsening, but not a tremendous amount, or maybe just staying the same. That's not what we saw. This looks like it just has symptomatic effects, maybe boosting or improving dopamine tone within the brain. So there is, the problem I mentioned is the word biomarkers. And so, this has been a priority area for Michael J. Fox Foundation and for the NIH, is to find ways to objectively measure these dopamine levels within the brain. And so if you could measure the actual dopamine cells, the integrity of these cells, a few years ago I showed you this particular promising biomarker. It's called free water. This is just an MRI scan that we can do and incorporate into clinical trials. And this is the part of the brain where you see these blue blotches where the dopamine cells are. And you can see here that this free water slowly increases in the brain from baseline to year 1, to year 2, to year 4. And it appears that this free water in these dopamine cells that we can measure on an MRI scan correlate with a patient's clinical symptoms. And so exenatide actually is now being studied, and the primary outcome measure is changes in brain MRI free water. I think some of the Parkinson's patients here may have enrolled in a study that we did here, a biomarker study that we did looking at free water, and some also may have enrolled in a study down at University of Florida that is also looking at free water using a different medication called rasagiline. That same group is looking at exenatide as well. That's a newer study that they just started. And so using biomarkers in these disease-modifying trials is key, because what we don't want is to come up with a positive study like exenatide, which is a quote-unquote positive study, but we still don't know, is it actually slowing the progression of the disease? So those immunization studies that I mentioned by Biogen and by the other company, Roche, they are having lots of biomarkers involved in their studies, so that if those studies do have positive results, we don't end up where we are with the exenatide trial right now. But those exenatide results are going to stimulate further bigger studies with biomarkers to see perhaps if exenatide is something that we should be looking at more intensely. So let me just finish up here before lunch, talk quickly about smoking. So, smoking is very interesting because we all know it's very bad for you, right? Nobody would ever recommend for you to start smoking. But like I talked to you about last year, smokers have this huge reduced risk of developing Parkinson's disease. When you look at smokers over time, there's a 77% reduced risk for developing Parkinson's disease. And that's what was the justification for the nicotine patch study that failed. The nicotine patch patients actually did worse than the placebo patch, so it's not nicotine. But I just want to introduce you to some work that I've been doing and some other people here at UB. We got very interested in lithium. Lithium is not even a compound, it's an element. It's in the earth. It's the third element on the periodic table of elements, which I think is the next slide. There you go. Helium, hydrogen, helium, and lithium. So lithium is found in very high concentrations in tobacco, over 20 times higher than any other plant or animal food that's been tested. And that was first showed in Indian tobacco back in 1980. A group in India showed this. And so we measured lithium levels in common Western cigarettes. I went to 7-Eleven and I bought a lot of smokes. And I brought them to my colleague, Murali Ramanathan, and he measured lithium levels. And so you can see in Indian tobacco, the level's 12, Camel Blue, it's 25, Camel Turkish Gold, 15, Marlboro, 15, 12. These levels are over 20 times higher than any other animal or plant food tested. Number 2 was black-eyed peas. That has a level of 0.6. So very high levels of lithium. I just am curious about lithium for a variety of reasons, but one of the main reasons that I'm curious about lithium is because there's a lot of animal data that lithium is neuroprotective, and again, it's the same story with many of our other repurposed drugs. They look good in animal studies, but how is this actually going to pan out in human trials? And so myself and a researcher at Beth Israel Deaconess, we've been working to try and get funding to do a lithium study in patients with Parkinson's. So far, we haven't been successful. But I just mentioned this last slide here. It's a busy slide. It reminds me of a bad Christmas outfit that went through the blender. There's lots of reds and greens, and you might not be able to see, but this is a paper that Dr. Andrzejewski, Dr. Lichter, myself, and a colleague of ours, Dr. Anderson, she's the Buck Institute of Aging, just north of San Francisco. These compounds, exenatide, lithium, urate, they share some similar mechanisms of actions targeting kinases. This is a paper that I expect is going to be accepted for publication this week. And I just mention it because it's kind of interesting that when many different compounds all seem to be targeting similar targets, in this case, kinases, increases the chances that maybe some of these are viable targets that we should be focusing on in our future research efforts. And you can, I'm not sure if you can see this, but this star right here is alpha-synuclein. And nilotinib will decrease alpha-synuclein, urate, lithium. This is all in animal models. Many of these targets, there's a lot of overlap. And so this is just some of the background that, if you're interested, is some of the kind of background that we can support future research. And so just to finish up, since we don't have a proven way to slow down Parkinson's, I hope you all have heard how important exercise is. We do have some evidence that daily aerobic exercise, Zumba classes, people have loved the boxing classes, moving, getting up and moving, the dance classes. This is one of the best things that you can do for your Parkinson's. It will make you feel better today, and we believe it can help preserve your future. So get up and move. Thank you.